Hey guys, Mr. Hyatt here. This is part 2 of the AP Biology Chapter 11 lecture on cell communication. So intracellular signals are going to be things that are located in the cytoplasm that receive a signal and can pass through a cell membrane. So they're going to be signals that are received in the cytoplasm that then move into the nucleus and affect. You see here we've got a receptor complex. It's going to accept expression. It's going to change proteins that are being produced. Kind of an exception to intracellular signals, or kind of a special type of intracellular signals, are plant hormones. Plant hormones can travel through the air or through liquid environments, like through a vascular system, like through veins and things like that. So their signals can travel through the air or through liquid. So that's kind of crazy. We definitely don't have that ability as animals. So again, an intracellular signal is going to get into the nucleus and turn on genes to manipulate types of proteins that are out. It's going to essentially turn on a protein that's needed to fix the problem, whatever the problem is, based upon the stimulus. Most signals never enter a cell. Most signals are received at the membrane, and then we use something else to carry the message into the cell. It's just a defense mechanism to keep things out that shouldn't be there. So how do we carry things from the receptor that lives on the membrane into sometimes all the way into the nucleus through a set of membranes? And then, how do we do that? Well, that's called a signal transduction pathway. So we're going to move the signal. We're also going to amplify the signal. We're going to make the signal louder. We're going to, instead of having one messenger molecule, now we're going to have 10 messenger molecules. Now it's going to have multiple steps most of the time, like protein kinases are going to be involved. There are going to be multiple relay proteins that pass things around. Here's an example. So we've got our signal molecule, we've got our receptor. There's an inactive protein kinase. That you should kind of know what that's going to do. Since it's a kinase, it's going to do something specific. Since it's a protein, it's made of something specific. So you should be able to word skills put together what that thing is. It doesn't matter which specific one it is. It's just some inactive protein kinase. We get a signal that activates the protein kinase, that activates the next protein kinase, that activates the next one. That's called a phosphorylation cascade because the activation is cascading down. Eventually, you're going to activate a target protein, or you're going to make it through the nucleus, and you're going to activate expression, something along those lines. Eventually, something's going to happen. The phosphorylation cascade is just turning on different messengers. Excuse me. So a protein kinase is a general name for any enzyme that's going to transfer phosphate from ATP to a protein. About 1% of our genes are for protein kinases, so that's pretty common. 1% doesn't seem like that much, but it's one thing, and we've got a crap load of DNA, so that's a pretty significant portion. Most frequently, we're going to phosphorylate serine and threonine. Remember, we're talking about proteins, so these are amino acids, but that's really beyond the scope of this class. In terms of amplification, I mentioned that we make the signal louder. Well, the protein kinase can often turn on multiple molecules. So we may get some branching where one signal sends 10 different versions of the same message. So we get an amplified response. So if we're trying to get a protein, if we need, say we've got a cut and we need to heal that cut, we need to patch that cut, we need to clot. Well, we don't need just one copy of the protein. We might need hundreds of thousands of copies of the protein. So the fact that we can amplify our signal and we don't have to wait and send a signal, okay, I need another one. Okay, I need another one. Okay, I need another one. That's a slow process. The fact that we can amplify that signal and we can say, hey, I need clotting factors, and we can produce a bunch of them all at once, speeds up the process, and it's definitely an evolutionary advantage. So then we get to termination. Phosphatases are going to inactivate the protein kinases, which is going to end transduction. Essentially, it gets a stop signal. So if we could alter the protein kinases in the cell cycle regulation, couldn't we slow the growth of cancer? Most likely. There's some pretty promising research going on in that field today. Again, it's not It's not a cure for cancer, it's just a way to circumvent it. Excuse me, sorry, I had to clear my throat again. Wanted to put this slide up just as our summary. This kind of shows a couple things. Let's just start over here on the signaling pathway. You see the reception, the transduction, and the response. So this is a real-life example of all of these molecules, what they're named. Excuse me, sorry. A few of their names anyway. And you see that we're just phosphorylating to activate these messengers, these second messengers. We're carrying that message. We're carrying it from point A at the membrane to point B, wherever that might be. Our response is going to be something like breaking down glycogen. That's the signal that we got. We had epinephrine bind, so that means break down glycogen, and we've done that. Notice over here that we've got our number of molecules activated. Quickly, it's going to grow exponentially. So we're activating a ton of molecules. From one signal, we get 10 to the 8th molecules. So we get a pretty big response from just one signal. I mentioned second messengers on the last slide. They're going to be small water-soluble non-proteins that pass on a signal. They're going to be spread rapidly. Because they're water-soluble, they're going to move around the water, and they're going to activate these relay proteins. Some examples are CAMP, which is cyclic AMP, calcium ions, and IP3. We'll talk more about that here in just a minute. So CAMP, or cyclic AMP, is a form of AMP, adenosine monophosphate, made directly from ATP. So we take an ATP, we take two phosphates off, and we connect it that way. It's not very stable. It quickly converts back to AMP once it's activated the kinases. Here's what I'm looking for. All right, here's what I'm talking about. So here's our ATP. We take two of those off. We wrap it, and it becomes cyclic AMP. Very quickly, that's going to revert that. Notice it's just one chemical bond that's breaking right here. That's going to happen spontaneously. Calcium ions are used much more widely than is cyclic AMP. Cyclic AMP is really important. Calcium is just more common. Use a second messenger in both G protein and tyrosine kinase reactions. Sorry. Really struggling with this phlegm. So yeah, here's an example of a calcium pump. We activate it, we pump. We activate it, we pump. So again, it's just going to carry the message from A to B. Calcium is going to be a transmembrane messaging a carrier, if that makes sense. It's going to work because of the difference in concentration. We got a concentration gradient. We're opening these channels and we're pumping them in. IP3 is a second messenger that is attached to the cell membrane. It works with calcium to do a lot of the same things that calcium was going to carry. Here's what I'm talking about. So we've got a G protein and a tyrosine kinase. Doesn't matter. Either way is okay. We could start here and follow this pathway to DAG and IP3, which is second messenger. Second messenger is going to carry the message to the ER. That's going to open the calcium channel. That's going to allow the calcium to diffuse in. Notice it's an IP3 gated calcium channel, kind of like a voltage gated, but it's just bound by the ligand to IP3. We can also use G protein coupled signaling to activate IP3 as well. Slightly different process. We're going to activate phospholipase C, which I guess is in both. Just different ways to get to the same end point. I kind of talked through this stuff. IP3 comes down. Calcium, the gate is open, so we pump calcium out. That's going to send our signal elsewhere, and it's going to activate proteins, and that's going to give us our response. In terms of responses, we could either regulate the cytoplasm or we could manipulate transcription. If we're messing with the cytoplasm, we're going to rearrange the cytoplasm, or we're going to open ion channels, kind of like you saw with IP3. We could also alter metabolism by turning different enzymes off, turning different enzymes on that are floating in the cytoplasm. If we're manipulating transcription, then somehow, some way, we're getting into the nucleus and we're expressing different genes than we would if we didn't receive that signal. Typically, those are activated by protein kinases. So yeah. Of course, here's a picture. I've got our growth factor, our receptor, activate, cross into the nucleus. We're going to have to activate a transcription factor, and that's going to express that DNA. So sorry. So keep in mind the specificity of our communication. Think back to when we played telephone. You're sending out a message. That message has an intended target. You don't want every cell in your body to start making clotting factors or to start breaking down glycogen. You want specific cells to do specific things. Well, different cells have different receptors. So we may need to couple things. We may need to use multiple receptors. We may have only one specific receptor, so that's the only cell that can receive the signal. Be like having a walkie-talkie instead of using a radio. There's only one person that can receive your message. An alternate explanation might be that the transduction pathways are different in different host cells, or different target cells, I should say. So you receive this one, you get a single response. If you receive the same signal, but the pathway branches because you have different relay molecules in here, you get 2 responses. Maybe you're activating or inhibiting something along these lines, and you get what they call crosstalk, kind of like negative feedback inhibition, something like that. And then if your receptor is different from these, then of course you're going to get a different response. We are focusing mostly on activating signals, but there are also inactivation signals that cause things to stop. But like I said, we're focusing on go signals, not stop signals, but don't lose sight of that. So we can typically use scaffolding proteins to hold proteins together. It's kind of like a lattice. You're giving your vines a place to grow up the side of your house. Same thing here. You're scaffolding these things together. Here's a picture. You've got a scaffolding protein that's going to hold these protein kinases so the receptor receives a signal. That activation pathway happens really fast because they're all right next to each other. This one turns this one on, which turns this one on, which does something else. Quickly to talk about apoptosis, it's going to be programmed cell death. So it's going to use cell signaling. DNA is going to chop up. The cell's going to shrink and become lobed. You'll see what that means on the picture. And then the pieces are digested by scavenger cells. So this sometimes happens in development. You see we've got this interdigital tissue. It's like webbing between our fingers. Those cells have to die at a specific time so that the webbing can be eaten away and we can form our fingers. Here's a white blood cell before and after. This is that blebbing that I mentioned on the 2 slides ago. So the white blood cell is starting to shrink and become lobed. It's just showing markers. Again, that's the balance between signals for stop and go, stay alive and die. Definitely going to be triggered by mitochondrial damage. If the cell realizes that you can't make energy anymore, that cell's going to automatically trigger apoptosis. Neighbor cells can trigger apoptosis. If there's a die signal nearby, that can cause cells to die, especially with Parkinson's and Alzheimer's and those glial cells and the Schwann cells that we talked about. And then internal signals like, hey, it's just time for that cell to go, things like that. There's a picture of the apoptotic receptor. You see that CED9 is inactivated, so these things start to break down. Proteases and nucleases start to break down the molecules. Got a video here that shows it. Check it out. Make sure you understand it for the test. Here's our summary. Don't get bogged down in the detail. Use the KISS principle. Keep it simple, silly. Know the 3 stages and at least one example of a receptor and how it works in detail, and make sure you understand G protein and the protein kinases and amplification cascades and things like that. Have an example of a secondary signal, a secondary carrier, second messenger, however you want to say that, and then make darn sure you understand apoptosis. That's chapter 11.